Hi, my name is Janae Heiss, and I am a student at Frostburg State University. I'm getting my master's in applied ecology and conservation biology. Before Frostburg, I went to Mount St. Mary's, getting my bachelor's in biology, and Hagerstown Community College, getting my associate's in biology. At Frostburg, my current thesis is on cannabis cultivar chemotype and genotype. Basically, that means different types of cannabis and how they look for the chemical composition and for the genetic composition. This ties into this video on cellular respiration because all things do cellular respiration, even plants, animals, everything. It's one of the fundamental concepts in biology. This video, per Dr. Putoff's request, is 60 minutes long and does not use slides or notes. This is a lecture on cellular respiration, which is Chapter 9 in a typical Bio 149 lecture series. Alright, so in this video, we'll be covering how organisms get food, how they break down that food, redox reactions, different forms of cellular respiration, how organisms make ATP, and then the 4 different stages of cellular respiration. All cells need energy to function. And cellular respiration is the way that cells get that energy. When you eat food, your body breaks down that food in your stomach. It's then absorbed and taken into your body to be used by your cells. The cells in your body break down the small molecules from your food, and they use it to make energy, carbon dioxide, and water. The molecule that's generally used for this reaction of cellular respiration, it's the model of all this that's broken down. And that molecule is glucose. Lots of other molecules can be broken down by cellular respiration too, but glucose is considered the model molecule for cellular respiration. In this video, I will be covering how different organisms get their food, how they break down that food, redox reactions, different forms of respiration, how ATP and NAD+ are cycled. Alright, let's try that again. In this video, I will be covering how organisms get their food, break down that food, redox reactions, different forms of respiration, the cycling of ADP to ATP, the cycling of NAD+ to NADH, and then the 4 stages of cellular respiration and the different types of respiration throughout the cellular respiration cycle. Before we get into cellular respiration, let's look at how organisms get food. All organisms are grouped into 2 categories based on the way that they get their food or their nutrition. And those 2 groups are autotroph and heterotroph. And both of those words are derived from the Greek. Autotrophs are animals or organisms, that make their own nutrition. The word autotroph is from the prefix auto, which means self, and then troph, which means nutrition. So autotroph, self, nutrition. Autotrophs make their own nutrition using photosynthesis. And examples of autotrophs are plants, cyanobacteria, and algae. Heterotrophs are organisms that have to obtain their nutrients from other organisms. These organisms are the ones that eat. The word hetero means something different or other, and then troph, again, means nutrition. So these guys use other nutrition. So when we're talking about heterotrophs, we're talking about things that use other nutrition sources than just themselves. 95% of all organisms are heterotrophs. Anything that eats plants or animals or anything are heterotrophs. And we are heterotrophs. So we've got food, but we need to break it down. When your food is digested, it's broken down into its basic parts: amino acids, fatty acids, and monosaccharides. In humans, the small intestine absorbs those monomers into the bloodstream and uses them to fuel chemical reactions throughout the body. Glucose is one of those monosaccharides that are being used for chemical reactions. Glucose is like this. It's a 6-carbon sugar that has the formula C6H12O6. It has a ring structure, and that's a hexagon ring. So cellular respiration takes all of these CH and CO bonds and it breaks them down to make energy. So we have the food, but we need to break it down. When your food's digested, it's broken down into its basic parts: amino acids, fatty acids, and monosaccharides. In humans, the small intestine absorbs those monomers into the bloodstream and uses them to fuel the chemical reactions in the body. Glucose is one of those monosaccharides that's being used for chemical reactions. Glucose, as seen here, is a 6-carbon sugar that has the chemical formula C6H12O6. It has a ring structure, like a hexagon. Cellular respiration uses the chemical energy stored in these bonds here, the CH and the CO bonds, to make energy. Cellular respiration has 4 main phases. In each phase of cellular respiration, the steps are all catalyzed by enzymes. So redox reactions are really important here. The enzymes take the electrons from one molecule and they transfer them to another. Electrons are transferred from one step to the next and they take energy with them. Redox refers to reduction and oxidation. So redox. So that's reduction and oxidation. When we look at that whole thing, redox reactions are reduction and oxidation reactions. In these reactions, one substance is oxidized and the other is reduced. Oxidation occurs when an atom loses electrons. So lose electrons, oxidation. And then reduction occurs when an atom gains electrons. So gain electrons, reduction. These reactions are always paired. In other words, for one atom to gain electrons, another atom has to lose an electron, and vice versa. So one atom gains an electron, the other loses an electron. Electrons aren't just floating around in space, they're on molecules. When glucose is broken down in cellular respiration, it is completely oxidized, so it loses all of its electrons. Those electrons have to go somewhere. And that's where reducers come in. Molecules that accept those electrons are called reducers. Some of the main reducers of glucose are NAD+ and ADP. NAD+ is short for nicotinamide adenine dinucleotide. When NAD+ takes on 2 electrons and a proton, which is typically annotated as a positively charged hydrogen ion, NAD+ becomes NADH. NADH eventually transfers the electron to a final electron acceptor and then becomes NAD plus again. Most energy in the cell is stored in the form of ATP. And here we have ATP. ATP is short for adenosine, which is this part, and then triphosphate, 3 phosphate groups. It's like the adenine in RNA, which would be that much, but instead of having 1 phosphate group, it has 3. Hence the triphosphate. ATP cycles between adenosine triphosphate and adenosine diphosphate, or ADP. It loses a phosphate group. Now, it stores the energy in the bonds between the phosphate groups. So, adenosine triphosphate has the most energy stored. ATP is considered to be the main energy currency for the cell. So when the cell wants to build up energy to do things like repair the cytoskeleton or bring molecules in through the cytoplasm, it uses ATP to do it. When we talk about cellular respiration, what we're talking about is how cells respire. When a person respires, also known as breathing, they breathe in oxygen and they exhale carbon dioxide. When a cell respires, we're talking about all of the cellular processes that involve the cell using oxygen as a reactant and making carbon dioxide and water as products. Energy is another main product of cellular respiration. All organisms need to have some mechanism to convert molecules of anything into ATP in order to make energy. But there are 3 different methods to do so. There are 3 main mechanisms that convert the molecules of sugar, protein, whatever, into ATP. And those are aerobic respiration, anaerobic respiration, and fermentation. Aerobic respiration is when oxygen is the final electron acceptor. Anaerobic respiration is when an inorganic molecule is the final electron acceptor. Anaerobic means it's without oxygen. So a molecule that's not oxygen is the final electron acceptor. And then lastly, fermentation occurs when the final electron acceptor is an organic molecule. Now there's more differences between respirations and fermentation than that, but those are the main differences in terms of electron acceptors. Aerobic, it's oxygen. Anaerobic, it's another molecule besides oxygen that's more inorganic. And then fermentation, it's an organic molecule. This video mainly focuses on aerobic respiration because plants use aerobic respiration. Additionally, most Bio 149 classes focus on aerobic respiration. The equation for aerobic cellular respiration with 1 molecule of glucose is this. So, we have 1 molecule of glucose, C6H12O6, and that is added to 6 molecules of oxygen, and it results in 6 molecules of carbon dioxide and 6 molecules of water. And that's the overall equation for aerobic cellular respiration. It's much more complicated than this, but this is the general equation for it. And this reaction has a delta G of negative 686 kilocalories per mole. So it releases a lot of energy. Breaking down glucose. it makes a lot of energy. But because if we took and got all of that energy at once, it would dissipate as heat. Cells have to take and get that energy in little bits. So they release that energy in small steps incrementally so that they can get that energy they need without losing it all as heat. Electrons in this pathway go from a high energy state to a low energy state, releasing a little bit of energy with each step they go. There are two main ways to make ATP in cellular respiration. The first way is called substrate level phosphorylation. In substrate level phosphorylation, ADP is present and then a phosphate is added from a neighboring molecule and it's used to make ATP. That molecule, also called the substrate because we're dealing with enzymes, it can be anything. But some enzyme is used to transfer a phosphate group from that molecule substrate onto ADP to make ATP. The second way that ATP is made in cellular respiration is through oxidative phosphorylation. And this way involves ATP synthase, which is an enzyme used to make ATP. It also needs a proton gradient, which provides that energy needed to produce the ATP. ATP synthase acts like a turbine. It uses the proton gradient to push the protons through and make the ATP from ADP. For aerobic respiration of glucose, there are 4 main stages. Those 4 stages are glycolysis, pyruvate oxidation, the Krebs cycle, and oxidative phosphorylation. So let's get into the first stage, which is glycolysis. Glycolysis is another Greek word. The first part of glycolysis is glyco, which means sugar. And then the second part is lysis, which means release or break. So in glycolysis, glycolysis, what we're doing is we're using sugar and we're breaking it. So we're breaking down or breaking apart sugar. In this case, the sugar we're using is glucose. Glycolysis occurs in the cytoplasm, and it's the only stage that occurs in the cytoplasm. The other 3 stages, pyruvate oxidation, Krebs cycle, and oxidative phosphorylation, they all occur in the mitochondria. Glycolysis is the only stage in the cytoplasm. Glycolysis is done so that it splits glucose in half, and it gives 2 3-carbon molecules, which are called pyruvate. Every single organism does glycolysis. At first, glycolysis is an endergonic reaction. Endergonic means energy consuming. So, it takes in energy to get started. It needs 2 ATP to get the reaction going. But by the end of the reaction, 4 ATP are produced. So it becomes an exergonic reaction. Exergonic means energy producing. So we produce 4 ATP. And overall, because we have a net gain of 2 ATP, glycolysis is an exergonic reaction. The full reaction for glycolysis involves 10 different steps, and each of those 10 steps are catalyzed by their own enzyme. And in higher level classes, they get into those steps. In glycolysis, there are 10 steps, and each of those steps has an enzyme. So we have 10 enzymes. Let's get into more detail for each of those steps. In step 1, glucose is converted into glucose 6-phosphate by the enzyme hexokinase. This step uses 1 ATP molecule. So right now it's an energy consuming reaction. Alright, then in step 2, glucose 6-phosphate is converted into fructose 6-phosphate by the enzyme glucose 6-phosphate isomerase. Then, in step 3, fructose 6-phosphate is converted into fructose 1,6-bisphosphate by the enzyme phosphofructokinase using 1 ATP. So far, we've got those places where those 2 ATP molecules are consumed during glycolysis. One in step 1 and one in step 3. Then in step 4, the enzyme aldolase splits fructose 1,6-bisphosphate into hydroxyacetone phosphate and glyceraldehyde 3-phosphate. So we have 2 different molecules here, but we want to make them the same. So then in step 5, triose phosphate isomerase is used to convert the dihydroxyacetone phosphate molecule into another G3P molecule. So we have 2 molecules of G3P. G3P is the product of photosynthesis. So in the next video, when we get into the Calvin cycle, the product of that Calvin cycle is G3P. In step 6, after we have those 2 G3P molecules, we convert them into 2 molecules of 1,3-bisphosphoglycerate, and 2 NADH molecules are produced by that. To convert G3P into 1,3-bisphosphoglycerate, we use the enzyme glyceraldehyde 3-phosphate dehydrogenase. And then, in step 7, 1,3-bisphosphoglycerate is going to be converted into 3-phosphoglycerate. So the 2 molecules of the 1,3-bisphosphoglycerate is converted into 2 molecules of 3-phosphoglycerate. And this conversion is done by the enzyme phosphoglycerate kinase. And this process makes 2 ATP, 1 for each molecule. So right now, we've got equal the energy made as being consumed. We have 2 ATP that were consumed in steps 1 and 3, and now we got 2 ATP made in step 7. In step 8, phosphoglycerate mutase converts the 2 molecules of 3-phosphoglycerate into 2 molecules of 2-phosphoglycerate. In step 9, enolase converts the 2 phosphoglycerate molecules into 2 molecules of phosphoenol pyruvate. And during this conversion, 2 molecules of water are produced. And then last but not least, in step 10, phosphoenol pyruvate is converted into pyruvate. So we have those 2 phosphoenol pyruvate molecules converted into 2 pyruvate molecules. And the enzyme that does this is pyruvate kinase. And then for this, 2 ATP are produced, 1 for each molecule. So this step makes it an energy producing reaction, because we've got those 2 more ATP that are produced in step 10 that push the total energy produced to 2 ATP. In short, glycolysis occurs in the cytoplasm. The net reactants here are glucose, ADP, and NAD+. So we use 2 ADP and 2 NAD+. And then, for every glucose, we've got net products. And those products are pyruvate. We make 2 pyruvate from every 1 glucose, 2 ATP, and then 2 NAD+, along with 2 positively charged hydrogen ions and 2 water molecules. And all the ATP produced here is via substrate level phosphorylation. So, remember from earlier, the substrate level phosphorylation is when we use some molecule as a substrate and take an enzyme to transfer that phosphate group onto ADP to make ATP. After glycolysis is done, the next step in aerobic respiration is pyruvate oxidation. Now this is the branching point where fermentation and respiration differ. And that respiration can be either anaerobic or aerobic. For respiration, organisms do pyruvate oxidation. Pyruvate oxidation breaks down that pyruvate molecule more, and it makes more NADH that can be used in further stages. In fermentation, the next stages of respiration with the Krebs cycle and the electron transport chain, they don't happen. So instead, those 2 NADH molecules that were made in glycolysis need to go somewhere. They need to be cycled back into NAD+, so that the organism can do glycolysis again. In fermentation, there is no oxygen needed. Some examples of fermentation are lactic acid fermentation, which occurs in yogurt, or ethanol fermentation, which occurs in yeast. But the focus here is on aerobic respiration. So let's go back to pyruvate oxidation. Pyruvate oxidation occurs in the mitochondria for eukaryotes, that organelle. And it occurs in the plasma membrane of certain bacteria, because they don't have organelles. Pyruvate is converted into acetyl-CoA. Acetyl-CoA. That CoA stands for coenzyme A. And it's converted into acetyl-CoA by the enzyme pyruvate dehydrogenase. Coenzyme A just acts as a link onto the acetyl group to give it stability for the next stage, which is the Krebs cycle. Pyruvate has a carboxylate group, and that carboxylate group is released into carbon dioxide during this process. We have an NADH+ that's converted into NADH during this process too. So we start with a 3-carbon molecule of pyruvate, and we make a 2-carbon acetyl-CoA, and we make carbon dioxide, and we make NADH. Because there were 2 pyruvates that were made during glycolysis, this happens twice. So we have 2 of each reactant and 2 of each product. So we have 2 acetyl-CoAs, 2 carbon dioxide, and 2 NADH. And no ATP is made during pyruvate oxidation. But because the focus here is on aerobic respiration, let's go back to pyruvate oxidation. Pyruvate oxidation occurs in the mitochondria of eukaryotes and the plasma membrane of certain types of bacteria. Pyruvate is converted into acetyl-CoA, and CoA here stands for coenzyme A, and it's converted by the enzyme pyruvate dehydrogenase. Coenzyme A links the acetyl group to give it stability for the next stage. Pyruvate has a carboxylate group, and this group is released into carbon dioxide during the process. Additionally, there's an NADH+ here that's converted into NADH. So overall, the reactants for pyruvate oxidation are pyruvate, NAD+, and coenzyme A. The products are 2 carbons, acetyl-CoA, and then carbon dioxide and NADH. There were 2 pyruvates produced during glycolysis, and so we end up with 2 of each reactant and 2 of each product. So just to remind everyone, there is no ATP produced during this phase. After pyruvate oxidation is the Krebs cycle. The Krebs cycle is also known as the citric acid cycle or the TCA cycle. The Krebs cycle takes the last two carbons from the acetyl-CoA and it breaks them apart. There's nothing left from the original glucose at the end of this cycle. The Krebs cycle occurs in the mitochondrial matrix, which is the innermost part of the mitochondria. There are 9 total steps in the Krebs cycle, and those 9 steps are broken down into 3 sections, like so. To start the Krebs cycle, the 2-carbon acetyl group from that acetyl-CoA molecule is added onto the oxaloacetate molecule, and those two come together making a 6-carbon molecule called citrate. This is why it's sometimes called the citric acid cycle, because you make citrate during the cycle. Now, the CoA from acetyl-CoA goes away during this time. After this first phase, where oxaloacetate and acetyl-CoA make citrate, then we get to the next section. And in this section, citrate rearranges and it decarboxylates. That's a fancy term which means it loses the carbon. So, we have the 6-carbon molecule and it loses a carbon dioxide to become a 5-carbon molecule, and then it loses another carbon dioxide and becomes a 4-carbon molecule. And in that process, it takes some NAD plus and it makes NADH. So, and this is happening for every acetyl-CoA. And we need to keep in mind that we made 2 acetyl-CoAs during the process of pyruvate oxidation. So this entire cycle is happening twice. So once we've got the 4-carbon molecule, we have the 3rd section. And this 3rd section is for the regeneration of oxaloacetate. And in that, we take that 4-carbon molecule and we get some FAD2, we get another NADH, and we regenerate the oxaloacetate. And this cycle goes around and around for every molecule of glucose. So, when we go and make that cycle, for every glucose that passes through that cycle, it makes 6 carbon dioxide molecules, 4 ATP molecules, 10 NADH molecules, and 2 FADH2 molecules. To get into more detail on the Krebs cycle, in the first phase, the 4-carbon oxaloacetate and the 2-carbon acetyl group come together to make citrate, a 6-carbon molecule, using the enzyme citrate synthase. And then in the second phase, citrate is converted into isocitrate by the enzyme aconitase. Then, the enzyme isocitrate dehydrogenase decarboxylates isocitrate in a process that also causes NAD+ to become NADH. So we make an NADH molecule in this step. Once isocitrate is decarboxylated, it loses a carbon dioxide, because that's what decarboxylate means. And it becomes a 5-carbon molecule called alpha-ketoglutarate. There we go. Alpha-ketoglutarate is then decarboxylated by the enzyme alpha-ketoglutarate dehydrogenase complex. This results in another NADH being made, a carbon dioxide being produced because it's decarboxylated, and then a 4-carbon molecule called succinyl-CoA that's remaining. At this point, there is nothing left of the original glucose. Because there's nothing left of the original glucose, this starts the final phase, which is the regeneration of oxaloacetate. Succinyl-CoA is converted into succinate by succinyl-CoA synthase. This step also makes an ATP, and then succinate is converted into fumarate by the enzyme succinate dehydrogenase complex, and this generates an FADH2. And then fumarate is converted into malate by the enzyme fumarase. And then lastly, malate is converted back into oxaloacetate by malate dehydrogenase. And this whole process makes one last NADH. We've finished the Krebs cycle. In total, every glucose that goes through the cycle makes 6 CO2, 4 ATP, 10 NADH, and 2 FADH2. This might seem like a lot of products, but unfortunately, it's not. Not when it comes to energy production. So, that carbon dioxide that was made during the cycle, it gets expelled as waste. And the NADH and the FADH2 need specific electron acceptors so that they're useful. So far, the only energy that the cell can use is just those 4 ATPs. And all of the ATP made so far has been made via substrate level phosphorylation. To make more energy, we need to go into the final step. And that's the one we've been preparing the products like NADH and FADH2 for. That final step is called oxidative phosphorylation. Oxidative phosphorylation is divided into 2 parts: the electron transport chain and chemiosmosis. In the electron transport chain, the cell takes those 10 NADH molecules and those 2 FADH2 molecules that it made during the last 3 stages, and it uses them in that electron transport chain. The electron transport chain is a bunch of different protein complexes that are stuck in the inner mitochondrial membrane. And those protein complexes have 2 main functions. The one function is to take the electrons from NADH and FADH2 and then to transfer those electrons through the chain. And then the next function is to pump those protons across the membrane into the intermembrane space. NADH and FADH2 are oxidized into NADH plus and FAD. The pair of electrons that are gotten from that are then passed through the electron transport chain through all those proteins, and then those protons from that are pumped into the intermembrane space. So each of those proteins pump protons across the membrane. The electron transport chain is why aerobic respiration requires oxygen. It's also why anaerobic respiration requires another electron acceptor, such as sulfur, carbon dioxide, or nitrate. You need some sort of electron acceptor once those electrons are done passing through the electron transport chain. So to get into the specifics of the electron transport chain molecules, let's look at them further. So those molecules are called complexes 1 through 4, but each of them have a more descriptive name, and each of them have their own important function. Complex 1 is also known as NADH, ubiquinone oxidoreductase. This complex takes a pair of electrons from NADH and oxidizes it to NAD+. So it's the complex responsible for oxidizing NADH back into NAD+ to go back through the rest of cellular respiration. So the electrons that it takes, it gives to complex 2. In addition to transporting electrons from NADH to complex 2, it also sends 4 protons across the membrane for every pair of electrons that it passes. Complex 2 is called succinate ubiquinone oxidoreductase, or succinate dehydrogenase complex. This complex takes the pair of electrons from FADH2 and it passes them along the chain. So we've got the complex that takes the electrons from NADH, and we've got the complex that transfers electrons from FADH2. This complex does not directly transport protons along the membrane. Complex 3 is also known as ubiquinol cytochrome C oxidoreductase, or cytochrome BC. This complex passes 2 pairs of electrons through it, and it transports 4 protons across the membrane. And then finally, complex 4 finishes the electron transport chain. Complex 4 is also known as cytochrome C oxidase. The 2 pairs of electrons go through this complex and are then joined to hydrogen ions and oxygen ions to make water molecules. So this is the complex that takes those electrons and uses it to make water. Additionally, 4 protons are transported across the membrane in complex 4. So for every NADH and FADH2 molecule that is oxidized by the chain, 12 protons are transported across the membrane and 2 molecules of water are made. ATP synthase is an important molecule that is next to the electron transport chain. It makes ATP during the process of the electron transport chain through a process that's called chemiosmosis. The protons have been pumped into the intermembrane space, and so that intermembrane space is really acidic. All those protons want to get out to the other side of the membrane, but they can't. And that's where ATP synthase comes in. ATP synthase acts like a carrier and allows the proton across the membrane. But there's a catch. Every proton that goes through ATP synthase provides the energy that's needed to add a phosphate group to the ADP and make it ATP. Those protons bind to the oxygen and form water. In anaerobic respiration, those protons would bind with another molecule like sulfur and produce another molecule. ATP synthase is like a molecular machine. It looks kind of like a rotary egg beater, but with one beater blade. And the proton makes the machine turn, and the ADP is forced into ATP. The movement of the proton gradient across the membrane is also called proton motive force, because it's really good at getting ATP to be made. It acts like a good force, bringing it all together. So the overall yield for aerobic respiration is 36 ATP per glucose for eukaryotes. We've finished aerobic respiration. So now that we've got that done, let's look at where all that ATP came in from. So overall, we make 38 ATP per glucose in bacteria and 36 ATP per glucose for eukaryotes. Eukaryotes make less because it costs 2 ATP to get across the mitochondrial membrane. Membrane transport requires ATP. So you need those ATP for eukaryotes. Bacteria, they don't have the membrane. So they don't need to spend those ATP. But when we do aerobic respiration, we have the stage glycolysis, where we make 2 ATP, and we also make 2 NADH, and then we've got pyruvate oxidation, which makes no ATP, but it makes 2 NADH. We've got the Krebs cycle, which makes 2 ATP, and it makes 6 NADH and 2 FADH2, and that gives us all the different sources we need to make the final ATP. So all that NADH and all the FADH2 gets used to make ATP. So we have total 10 NADH, which translates into 30 ATP. And we've got 2 FADH2, which translate into 4 ATP. So when we take it and we go through each stage, we get 2 ATP from glycolysis, we have no ATP from pyruvate, we have 2 ATP from the Krebs cycle, and then we get that 34 ATP from breaking down the NADH into NAD+, and breaking the FADH2 into FAD. So we get all that ATP from the electron transport chain and from chemiosmosis. And that's why the electron transport chain is really important in getting a lot of ATP. So that's aerobic respiration. Now there are also other things besides aerobic respiration. There's anaerobic respiration and there's fermentation. Anaerobic respiration, remember an without, anaerobic, oxygen. So anaerobic respiration is similar to aerobic respiration, but there's another molecule besides oxygen that's used as the final electron acceptor. Now bacteria and archaea use many ways to get energy, and a lot of them use anaerobic respiration. Some hot springs bacteria use sulfur as the final electron acceptor. Now fermentation, it uses an organic molecule as the final electron acceptor. But it doesn't have an electron transport chain, and it doesn't have the Krebs cycle or pyruvate oxidation. It just has glycolysis. An ethanol fermentation uses yeast and breaks down that glucose into pyruvate and then down into CO2 and ethanol. So we've got glycolysis and then we've got ethanol fermentation. Another form of fermentation is lactic acid fermentation, and that breaks pyruvate down into lactic acid. Unfortunately, fermentation is not very efficient. The energy yield for fermentation is much, much less than aerobic respiration. Fermentation makes 4 ATP, just 4. Aerobic respiration makes 36 ATP in eukaryotes. That's like 9 times the amount of ATP. And so the energy payoff for aerobic respiration is much, much more than fermentation. And that is why more complex organisms tend to do aerobic respiration. They need that energy for so many other things. And there are different types of anaerobes. So we're talking about different types of organisms that might not use oxygen. So anaerobes without oxygen. Obligate anaerobes are things like bacteria. And obligate anaerobes need to be without oxygen. They can't survive in oxygen. They can do fermentation or anaerobic respiration, but oxygen kills them. Facultative anaerobes are yeast and bacteria. They are facultative. They can do it with or without, depending on what's available. These guys can do aerobic respiration, they can do anaerobic respiration, they can do fermentation. They've got the widest array of different types of ways to get their energy. Facultative anaerobes are really great at finding ways to get energy. And as stated earlier, just to conclude, the cellular respiration pathway is not only for glucose. There are so many different types of sugars, proteins, and lipids that can go into cellular respiration too, and they can be broken down to get energy. Different molecules can go into different parts of the cellular respiration pathway. They don't have to just start at the beginning at glycolysis. So if there's a G3P molecule that comes in there, they can go in a later step. And cellular respiration is not static. So it doesn't just go in that order. There are different variations to it, and it can regulate different parts of the cycle as needed. So the cell can make more ATP when it needs to make more ATP. So to conclude, cellular respiration is an important cellular pathway that organisms use to get the energy that they need from the food they eat. Let's look at some current research in cellular respiration. So these are some interesting things that happened this year. The first study is one on PPR proteins that modify the mRNA. Because plant cells obtain their energy from chloroplasts in the mitochondria, and we looked at the mitochondria here with cellular respiration, we see that the plant genes in the mitochondria are really important to that, because those plant genes can have mutations. There's an enzyme called the RNA ediasome that helps to repair those mutations. So there's an error correction in plants that involves converting cytosines from mRNA into uracil to correct the errors in the chloroplast DNA or the mitochondrial DNA. And this RNA editing is really important for processes like cellular respiration. So we have this group of proteins, and they're called PPR proteins, and they have DYW domains. And they are some of the main proteins for RNA editing. These proteins are transcribed in the nucleus, and then they get transported to the mitochondria, where they do their work. They're inactive during the transport, and they only become active after they go to the organelles. And up until now, people weren't sure how these proteins actually became active. But this year, they figured it out. A German-Japanese team of structural biologists and biochemists figured out how these proteins get activated. Dr. Gert Weber, Dr. Mizuki Tanaka, Tatiana Bartel, and Dr. Manfred Weiss were the main researchers on the project. They determined that there's a 3D structure of this DYW domain, and they figured out what shape it's in and how that shape works. And from there, they figured out a mechanism of DYW domain activation in plants. So a zinc atom in the center of this domain acts as a catalyst to accelerate how they get the cytosine to go to uracil. And they found that there was a gating domain in front, like around that zinc atom, that determines whether the zinc is available for reactions or not. And this gating domain can act like a switch to turn the enzyme on or off. The PPR proteins are normally inactive, and they receive activation that they need as they travel to the mitochondria and the chloroplast. This is a really good design, because if they were active on the way there, they could cause all sorts of damage to the cell and to the surrounding membranes and all of that. So this work describes a breakthrough for plant molecular biology, because it describes an additional level of gene regulation in chloroplasts and mitochondria. Another study from this year involves trying to look at how the electron transport chain is for plants. So there are differences at the molecular level between plants and animals, and how they extract their energy from food sources. And discovering those differences can help revolutionize agriculture. So this Maldonado and Letts and other researchers gave a atomic level 3D structure of complex 1, the one complex in the electron transport chain for plant mitochondria. They had structures for this complex in mammals and yeast, but they did not have anything for plants yet. And they hoped that by determining the structure of these complexes in the electron transport chain, they could improve pesticides. Because at this point, a lot of pesticides target the electron transport chain. So if they can get specific electron transport chain targeting to that pest without getting the plants, then we could actually have more specific pesticides that don't harm the plant and don't harm other organisms. So, because of their similar size, the researchers had problems isolating mitochondria from chloroplasts. So they had to actually isolate them by the mitochondria by growing the plants in the dark so that the chloroplasts wouldn't develop. This is pretty cool. So they purified the mitochondria and found the complex, and then they determined how it was assembled. And they actually found a potential assembly intermediate in complex 1 that was unique to plants, and that may have its own separate function. So looking at this is crucial for doing further plant research. By determining specific things about the electron transport chain in plants, we can not only figure out how to grow plants better, but we can also provide pesticides that would be more target specific. Having the plant respiration be at the optimal level is the best thing for plant growth and biomass accumulation. So with the unique structure of complex 1, researchers aim to determine how those functions of complex 1 exist and how they are different than the normal functions of complex 1. And they want to see how those functions can be applied to agriculture. The different research that we have for cellular respiration now is still ongoing, and it provides some amazing discoveries about plant biology, and there's also some animal biology stuff too. So there's a lot of different studies out there, and people are still doing ongoing cellular respiration research. Can we stop now? Thank you for watching this video. I hope you learned something about cellular respiration and how important it is for everything on Earth.